Hello everyone and welcome to our Flash Talk, visualization and graph exploration of the fruit fly brain datasets with NeuroNLP++. I am Mehmet Kerem Türkcan, a PhD candidate at Columbia University in the Department of Electrical Engineering, and this is joint work with my advisor Aurel Lazar and Ian Zhao. In this presentation, I will be highlighting our recent work on building a portal for navigating the fruit fly brain circuit data that's powered by a free-form question-answer engine, integrating neuron types in the fruit fly brain together with relevant literature. Recent releases of large-scale connectomics/synaptomic datasets have enabled experimental and computational neuroscientists to explore neural circuits in unprecedented detail. To efficiently explore these datasets requires, however, knowledge of both the biological nomenclature and programming tools. These skills are often limited to members of their respective communities, such as neurobiologists and computer scientists. To close the programming gap, NeuroNLP has enabled highly sophisticated English queries of Drosophila or fruit fly brain datasets. NeuroNLP, which you see here, is a browser application that provides a modern web-based portal for navigating fruit fly brain circuit data, specifically neurons and their synapses in 3D. The NLP in the name comes from the fact that our search engine is powered by a rule-based natural language processing engine. However, the NeuroNLP rule-based query engine can only map very specific, pre-designed sentence structures into database queries, thereby limiting its usage. An example is a sentence such as show glutamatergic local neurons in the left antennal lobe, for which the result I have just brought to our workspace. Deviation from preset sentence structures like this one cannot be interpreted correctly under this regime. Here, by clicking this button, I'm showing some of the examples of the query rules that we have enabled. Whereas useful, these queries require a good knowledge of the search manual that you see here and the neuron type keywords used in the dataset of interest. To enhance the user experience when asking questions that are well beyond the current capabilities of NeuroNLP, we devised the NeuroNLP++ Brain Explorer. To integrate Drosophila ontology datasets and other descriptive terms with connectomics/synaptomic datasets, NeuroNLP++ employs modern advances in document retrieval. NeuroNLP++ integrates cell types or lineages and matches them against neurons in a connectomics dataset. These descriptions and the corresponding neurons can then be queried using natural language sentences, even via questions that are difficult to anticipate using rule-based NLP queries. Simple examples include what types of local neurons are there in antennal lobe, or which neurons are known to respond to carbon dioxide. Here, I'll give you a short introduction to NeuroNLP++, which you see here. NeuroNLP++ includes a set of demos that can be used to learn about how to use it. I will now run one of these demos. The first demo you see when you open up the demos menu, as you see here, is about understanding how to use freeform queries. Let me click on it and run it. We run a simple query, such as, what are patchy local neurons? Which is about to come up here, right here. What are patchy local neurons? Which is a natural question to ask about a cell type called patchy local neurons. And the rule-based engine cannot easily give answers to this question. However, NeuroNLP++ can find answers with ease. On the left menu here, you will see various different matches that were made for this question. And as you can see, the first one was the adult patchy antennal lobe local neuron cell type. And clicking on the Add to Workspace button, I was able to bring these neurons, of which there are 14 of them, to my workspace. Secondly, NeuroNLP++ can be used when we have a keyword and want to get some more information about it. For example, let us say we know what Kenyan cells are and are trying to find Kenyan cell subtypes. Let me now run, by going here, the third demo we have, which is about such a case. In the third demo here, we ask for Kenyan cell subtypes by writing, what are Kenyan cell subtypes? And NeuroNLP++ returns a list of various entries associated with Kenyan cells, which can be used to discover subtypes we don't know about or to bring them to our workspace. For example, let me click this to bring alpha, beta, posterior Kenyan cells to my workspace. From the technical side of things, NeuroNLP++ uses dense passage retrieval and bidirectional encoder representations from transformers, BERT, specifically the PubMed BERT model, to find possible answers to questions pertaining to a collection of ontology terms and their descriptions. After the relevant ontology terms have been identified, NeuroNLP++ retrieves neurons matching specific names in the hemibrain dataset with the ontological terms and their synonyms. These matches are then brought into the 3D environment of the user's workspace, in which the synaptic partners of the individual neurons can be visually explored. To sum up, NeuroNLP++ empowers users to effectively and efficiently construct circuits and enables researchers without deep knowledge of nomenclatures used in connectome/synaptome datasets or programming skills to explore the brain circuits of interest. Thank you for watching, and I'd love to answer your questions or tell you more about our work. Thank you.